Assalamualaikum warahmatullahi wabarakatuh. My name is Luqman Hakim bin Abdul Halim from Fisheries. So today I want to present about the DNA sequencing. In this slide, I have four content, which is introduction of DNA sequencing, the Sanger method, disadvantage and advantage of the Sanger method, and application of DNA sequencing. So DNA sequencing is the process of determining the sequence of nucleotide within a DNA molecule. Every organism DNA consists of a unique sequence of nucleotide, which means even human we have different sequence of nucleotide. So determining the sequence can help scientists compare DNA between organisms, which can help show how the organisms are related. So this means the strength of DNA, it have four nucleotide bases, which is adenine, guanine, cytosine, and thymine. So these four bases are related to each other, and it has their own base pair. So this is the four base pair in the DNA. Adenine pair with thymine, cytosine pair with guanine. So adenine and thymine, it has 2 hydrogen bond. Otherwise cytosine and guanine, it has 3 hydrogen bond. So the difference between these two pair is it has for cytosine and guanine is strongest than adenine and thymine because it has advantage of 1 hydrogen bond. So for the method, the Sanger method, also known as a chain termination method, is a method for determining the nucleotide sequence DNA. The method was developed by two-time Nobel laureate Frederick Sanger and his colleagues in 1977, hence the name the Sanger sequence. Okay, for the step of Sanger method, we have four. First, denaturing of DNA. Second, additional DNA primer, DNA polymerase, special modified deoxynucleotide triphosphate, and free nucleotide deoxynucleotide triphosphate. So in the second step, we have to make sure each of the tube have normal and base, deoxy and base, which is deoxy adenosine 5-phosphate, deoxycytidine 5-triphosphate, deoxythymidine 5-phosphate, and deoxyguanine 5-triphosphate. So between these four, it separate into two groups, which is purine and pyrimidines. So in the purine group, it contain the DD-ATP and DD-GTP, which means it has a double ring structure. So pyrimidine, DD-CTP and DD-TTP, which has a single ring structure. So after the second step is done, we have to do electrophoresis that cause the DNA separate based on their size. After that, the sequence read from the bottom of the plate. So for the advantage, there will be no premature termination as a result of DNA sequencing. Second, identify the gene causing genetic disease. So we can identify the disease that causing by the gene in the animal or human. So third is widely used for 3 decades and even today for single or low throughput DNA sequencing, and for allow scientists to determine genome species. For disadvantage, not widely used and use radioactivity and toxic chemical. So it's expensive and time consuming. Third is low speed analysis does not follow the sequencing of complex genome such as plant species genome. So for the fourth is DNA sample misinterpret for crime investigation. Okay, for the second disadvantage, it may be expensive and it has a long time to do the PCR DNA sequencing. So better we do the another way to make the DNA result. So for the application of DNA sequencing, we have to establish evolutionary links within and between species. Homologous DNA sequence for distinct organisms can be compared. So we do the DNA sequencing to compare animal before this and animal that live in the century. So we know about the sequence of DNA animal. So we can know the species, the genus, and where it comes. So second is a gene sequence functional section can be investigated. Various domains that are common to protein with comparable function can be identified to establish the function of a gene. Okay, thank you.